Hi folks, I am Adi Aksanugraha.
And my name is Devani Putra. My name is Muhammad Rizky Aryoso.
And I am Fairuz Asharabani.
We are from
Eureka team.
We are from Sman School Bogor. Right here, we designed a project to help people get clean water more easier. But why and what we choose this project? Okay, let's do the video.
Okay. Clean water is a primary need of every living thing, and rivers are an important component in providing water for every living thing. There are many rivers in Indonesia, but unfortunately, rivers in big cities currently pollute, many of which contain microbiology and heavy metals. One of the heavy metal contaminants, lead, can be bad for human health. If this heavy metal is consumed, it can cause several complications, such as bone disorder, nervous system, kidney, heart, and even cancer. This is a threat to people who live along the river banks. Polluted rivers such as the Ciliwung River, Cisadane River, Citarum River, and Serak were recorded in the dry season to have an average lead contamination level of around 58.67 microgram per liter. The highest data belong of the Ciliwung River, which reached 140 microgram per milliliter and 136 microgram per milliliter during the rainy season. This level of lead contamination is far from safe threshold of 0.01 until 0.05 microgram per milliliter. The number of polluted rivers in big cities has reduced access to clean water for the community, which we know if water is the main need for every human being. From there we took the initiative to take this project as a step of change in order to provide access to clean water to many people. We named this project Red Eureka, reduce lead through genetic engineering bacteria. In our project, we designed bacteria capable of reducing lead. We use Ecoli DH5 alpha bacteria as a medium of propagation, which plasmid PSB1C3, which interests the SMTA gene from Staphylococcus pcc 7942.
So, how to reduce lead content from water? We have designed a genetically engineered bacterium using the principle of synthetic biology. In this presentation, we will explain the results of the project that we have been working on since last summer. So, we use Escherichia coli bacteria to engineer the plasmid. What is Escherichia coli bacteria? Escherichia coli or E. coli is a type of bacteria commonly used in genetic synthetic biology. It is a well-studied organism with a well-understood genome and is easy to grow and manipulate in the laboratory. This makes it an ideal host for engineering new biological system. Therefore, we engineered for one of its organelles, more precisely a plasmid to be inserted with plasmid PSB1C3. Plasmid PSB1C3 is a high copy number plasmid commonly used in synthetic biology. Okay, and it is derived from plasmid PMB1, which is natural plasmid first isolated from Escherichia coli. Plasmid PSB1C3 has several features that make it suitable for use in synthetic biology, including high copy number, a chlorophyll resistant gene, and standard biobrick assembly car, which allows to be easily combined with other biobrick parts. Okay. These plasmids are versatile tools that can be used for a variety of purposes in synthetic biology. It is commonly used as a backbone for the construction of new genetic circuits, as a cloning factor, and it also has been demonstrated and characterized by many testers. Many IGN teams have used this plasmid in their design. After that, we added a target gene that is an important part of our project goal, the SMTA gene. and which function as a lead reducer in water. Okay, what is SMTA gene? SMTA gene is a protein. It's like a small protein consisting of only 70 amino acids. It has a high affinity of metal ions and can bind a wide variety of different heavy metals, including zinc, copper, and special lead. The SMTA gene is taken from bacterium Synecopus PCC 7942. This gene has the ability to bind and reduce lead in water by converting it into a harmless compound like a PBO. Therefore, we add the ribosome binding sites RBS B0034. Okay, RBS is a short sequence of nucleotides located upstream of the start codon of an mRNA transcript. The RBS is responsible for reverting the ribosome to the mRNA and initiating translation. In synthetic biology, the RBS is used to control gene expression. By engineering the RBS sequence, scientists can control the amount of protein processed from a gene. Then, why do we use RBS B0034? We use this part because this RBS has been characterized and has the ability to support our plasmid design process. And in prokaryotic cells, RBS plays an important role in regulating gene expression. These RBS are essential elements of mRNA molecules that regulate translation initiation, which ultimately determines protein production. Their ability to influence protein expression levels makes them a valuable tool in the genetic engineering and synthetic biology. And for the promoter of this SMTA gene, we use the promoter part TBA1721001. This part is designed to allow lead detection in cells. When iron lead Pb+2 enters the cell, it will pair with lead binding protein, which is useful to activate the target gene. SMTA like to start the transcription process to reduce lead. In addition, this gene has also been used by previous IGEM teams, so its ability has been proven and tested. After that, we also use the terminator of the gene. The name is terminator B0015. We use this part because it has a better ability to stop the process after transcription in the gene compared to others, and also this gene has been characterized and tested. To insert the plasmid design that has been made into Escherichia coli, we use the ways, namely through the electrophoresis method by utilizing electrical energy to open the pores of the bacteria, and also chemical reagents using carbon chloride compounds by utilizing the difference in electrogradient concentration. Bacteria that have been inserted into the PSB1C3 plasmid will have a new ability to reduce lead. The process begins when the engineered E. coli is placed in a polluted environment that contains a lot of lead. The BBA1721001 promoter will immediately detect and begin to activate and send signals to the SMTA gene to start the transcription process. This transcription process starts at the origin of the replication. Therefore, replication cannot start randomly at any point in DNA. ORI is detected by initiator protein DNA in prokaryotes. Once ORI is located, helicase unzips this double strand by making replication fork. This process aims to convert iron lead Pb2+ into a harmless compound like PBO. When RNA polymerase reaches the terminator, it will automatically stop or deactivate the transcription process.
Discuss about our project. Indonesia, which a developing country, is still prone to problems with the living standards of its people biologically. But in our subject matter, it is about how to solve the lack of availability of clean water for humans. Therefore, we designed a plasmid by uniting the SMTA gene with the PSB1C3 plasmid and Escherichia coli, or we talk about E. coli. Okay, the unification aims to reduce the lead levels in water in order to increase the level of clean water at the source and make the problem of water containing lead can be resolved. Okay, that's for all.
We have a series of long discussion about this project, starting with determining the direction and purpose of our project, how our project can be implemented in the real world, what parts we will use designing our project, and how we use these parts so that can be united into a solution that can be used of our problem solver. From this long discussion, we have found answer and decision from the question above, including first, what is the purpose of our project? As already explained in the project background, we have determined the direction of our project, which is to design a bacteria that are able to degrade lead in water to help people in obtaining clean water, especially people who still rely on Ciliwung river water for their daily needs. 2. How can our project can be implemented? With in silico method, based on data search and information collection through credible journals and sources, we compile this project carefully and pay attention to all aspects of that possibility and existing knowledge, so that this project can really be done and tested in the real world. 3. So what did we use in designing the plasmid in this project? Through data collection from various sources and also iGenPart.org website, we have decided to use these genes as promoters, RBS, target genes, and also terminators in our plasmid design, which include A BBA1721001 as promoter. This is because this part is designed to enable lead detection in a cell. When lead or PB2+ enters the cell, it will couple with the lead binding protein, which is useful to activate the target gene, SMTS, to start the transcription process to reduce lead. In addition, this gene has also been used by previous iGEM teams so that its ability has been proven and tested. B. RBS, we use B0034. We use this ribosome binding site B0034 because this RBS has been characterized and has the ability to support our plasmid design process. C. Target gene, we use SMTA. This SMTA gene is taken from the bacterium Synococcus PCC 7942. This gene has the ability to bind and reduce lead in water by converting it into a harmless compound, or PBO. D. Terminator, we use B0015. We use this part because it has better ability to terminate the process after transcription in the gene compared to the other. And also, this gene has been characterized and proven in this ability. E. Plasmid, we use PSB1C3. We use this plasmid because it has been proven and characterized by many testers. Many iGEM team had used this plasmid in their design. And the last, media spread through E. coli DH5 alpha bacteria. Why this bacteria? This is because E. coli DH5 alpha is a good genetic engineering medium and can be said to be perfect when looking at the safety aspect and also the mechanism of action. In addition, this bacterium has also been characterized and has also been widely used by researchers including other iGEM participants in their genetic engineering design. Question number 4. How we combine the above parts? Okay, we compiled the tested the above part through the Benchling website. What is Benchling? Benchling is an online platform to connect the entire life cycle of research and design project, from the preparation of project reports to the pre-lab testing. To unite the above parts, we need to pay attention to each primer forwards and the primer reverse of each part so that the plasmid design it has been designed can work well and as expected. Melting temperature and also the bond between guanine and cytosine must be considered carefully. The result of our project development after a series of discussion and information search will produce a mechanism to able degrade lead in water through engineering Escherichia coli DH5 alpha bacteria. The Escherichia coli DH5 alpha bacteria that have been modified with plasmid PSB1C3, which is contains the SMTA gene from the Cyanococcus PCC7942. The gene is able to reduce lead up to 99%. Searched by bioremediation of lead by Zhang et al. And not only that, so that the engineered bacteria can work as expected and not become a boomerang for the community, we made a filtration system consisting from 3 reservoirs and 2 drains. Why do we need to implement the filtration system? Based on data and information we have done, if water contains massive amount of E. coli DH5 alpha bacteria, the water will be dangerous if consumed by living things, especially human. Therefore, if we directly implement the engineered bacteria directly into the river, there is a high possibility that the bacteria will develop uncontrollably, which will endanger life in water and maybe in the land. That's why we made this water filtration system. The filtration system consists of 3 reservoirs. The first reservoir contains water directly from the river. The water is still polluted and contains a lot of waste, especially lead. In this first reservoir, also the E. coli bacteria that have been engineered work. The bacteria will perform the lead reduction process in the first tank. After that, the lead in the water is reduced to several compounds. The water now has a high concentration of E. coli, which is dangerous condition. Therefore, the water then flows to the second reservoir, which is contain layers of natural filtration like sand, grave, pine sand, palm fiber, charcoal, cloth, and gravel to filter out E. coli bacteria, which are then cancelled to the third reservoir. In the third reservoir, water that is free from lead and E. coli bacteria is collected. So that's how our project mechanism work, and maybe I will going to the next part with my new friend.
Okay, after the explanation before, we talk about the conclusion about this project. In this project, we are using plasmid PSB1C3, which will be designed to contain the SMTA gene that encodes an enzyme to reduce lead. The enzyme will oxidize lead into PbO. The designed plasmid will be expressed in Escherichia coli or E. coli bacterial cells. The bacteria will absorb the lead from water and reduce it into harmless compounds. The results of this project are expected to be an effective solution to the problem of lead pollution in drinking water.
In additional information for solving local problem, we use computational biology to identify a design a lead reducing gene, the SMTI gene from Cyanococcus bacteria. We then clone the SMTI gene into the PSB1C3 plasmid, which is a standard for synthetic biology. We then inserted the PSB1C3 plasmid containing the SMTI gene into the E. coli DH5 alpha, a strain often used for genetic transformation. We used antibiotic selection to ensure that the E. coli bacteria that successfully took up the PSB1C3 plasmid were resistant to chloramphenicol. We used the modified E. coli bacteria as the dispersal medium to reduce the lead content in river water. We also made a water purification system consisting of a filter, tank, pump, and sensor to drain lead contaminated river water into a tank containing modified E. coli bacteria. The water that comes out of the tank is then filtered again to remove bacteria and other particles so that it becomes clean water that is safe to consumption. This we have shown how synthetic biology can be used to tackle local problems in an innovative and effective way.